Hey, welcome everybody. I think we'll go ahead and get started. First, I just want to make sure to tell everybody where they are. You're at the FBRI Maurice Strauss Distinguished Public Lecture. Like they say on airplanes, if you're flying to Chicago, stay on this plane. If you're not, get out, but don't anybody leave. You're at the right place. And as always, we want to, of course, acknowledge and thank Mr. Strauss and his family for the support of this program. Before I get to introducing tonight's speaker, I want to do what I always do, which is just give you a heads up about upcoming lectures. So turns out our March lecture in this series has been postponed due to the speaker having to change his calendar, but we're going to make up for it. We actually have two speakers in the month of April. On April 4th, we have Dr. Zulfikar Bhutta from the Toronto Hospital for Sick Children, who will be speaking on climate change effects on health of vulnerable populations. And then immediately after the next week, on April 11th, we'll have Dr. Kristen Yaffe from the University of California, San Francisco, speaking on progress in Alzheimer's disease diagnosis and treatment. So we have a good full program in the following month, even though we're going to have a gap of a month coming up. So it is now my pleasure to introduce this evening's speaker. Our speaker is Dr. Shelley McGuire. And before I get into the details, I want to say that Dr. McGuire flew from Moscow, Idaho, nonetheless, through someplace else in the Northwest, Portland, to Charlotte, to Roanoke yesterday. I don't know, it was like 11 hours or 13 hours. And then for her day to start this morning with meetings, we started her at 8 o'clock, which of course is 5 a.m. Pacific Coast time, meaning she got up at 4 a.m. Pacific Coast time after flying all day and all night. And she's still full of energy and going and so forth. So I'm duly impressed. Oh, you took her for a walk too. Good. Okay. Wake her up. But anyway, I'm very grateful for her spending the time to get here. It's a big trip, I know. Anyway, Dr. McGuire currently serves as the director and professor of the Margaret Ritchie School of Family and Consumer Sciences at the University of Idaho. She did her undergraduate work and her master's degree in nutrition science at the University of Illinois in Urbana-Champaign, where I used to be. We had some good discussions about that today. And then she did her Ph.D. in human nutrition at Cornell University after that. She joined the faculty at Washington State University and spent, I think, close to 20 years in that position, became full professor there. And then about 5 or 6 years ago, moved, I'll explain in a minute, to the University of Idaho. As it turns out, she didn't actually move. She just rode her bike this way versus that way to get to the new university, which is very close. Two land-grant universities within, 7 miles of each other. I had no idea that situation existed, but nonetheless, made an easy move. Dr. McGuire has received a lot of awards and recognition for her work and performs a lot of national service in her areas of expertise. For example, she won the International Life Science Future Leader Award, the American Society for Nutrition Excellence Award. She was elected to the National Academy of Medicine about a year and a half ago or so. She's been a co-leader on the NIH project on developing dietary guidelines for Americans, and one of the leaders of the U.S. Federal Human Milk Composition Database Project. She also chairs the National Institutes of Health Working Group on Human Milk Composition as part of the Breast Milk Ecology Infant Nutrition Project. And she advises Bill and Melinda Gates Foundation on International Milk and Lactation Consortium. So as you can see, she's highly regarded and recognized and contributes in many ways beyond her individual research at a national and really international level. She's published over 100 scientific peer-reviewed publications and 7 textbooks. Her work has provided important insights into the beneficial effects of specific fatty acids on human health, on milk micronutrients in lactating women, bacterial communities in human milk, obesity prevention, and the role of fast food consumption and human milk composition. More recently, she has been working on mapping the human milk microbiome in diverse human populations and studying the relationship in the microbiome and childhood malnutrition. And she's been also involved with work to study the safe use of bioactive ingredients in infant formula, the role of maternal nutritional status on human milk immune factors, and the role of breastfeeding pattern on human milk microbiome composition, as I said. So as you can see, she's truly a leader, a major contributor in this important field. And I think, as I said at our last talk a month or so ago, how can anybody not be interested in this? Who isn't interested in learning about milk and the formula and the nutrition that our children get? And somebody said to me at the last talk, well, our children are already grown up. Okay, your grandchildren. All right, if not your grandchildren, your neighbor's grandchildren, whatever. We're all interested in this. We greatly look forward to Dr. McGuire's talk. Please join me in welcoming her tonight.
Okay. All right, thanks everybody for having me. I appreciate the introduction. I need to get a little participation here. So I'm curious, how many students do we have here? Okay, mostly students, very good. How many faculty and clinicians? Ever? Very good. Community members? Excellent. Okay, community members, if I say something that doesn't make sense, please stop me. Everybody else, you should understand everything. I'm just joking. But I'm very open to being stopped, so please do that. So I'm really happy to be here today. I am going to be talking about the human milk microbiome. I really consider this a paradigm shift in infant nutrition and how what we think about how babies should be fed. And I'll talk more about that. I subtitled this A Lesson in Serendipity because as you see, I've never even had a microbiology class. I am not a microbiome person, but here we are doing microbiome by chance. So I'll talk a little bit about that. Okay, so what I'd like to go through today is just a little outline. I'm going to talk a little bit about Idaho and the University of Idaho because as we just heard, it's 7 miles away from another land-grant university, and I just want to give it a little bit of due. It's a really wonderful university. We're going to talk about how we got into the milk microbiome field. We're going to talk about what bacteria are in milk, a little bit about variability, nutrition, geography, caregiving networks, a tiny bit about so what, because we actually don't know. And then finally, I'm going to finish talking about COVID-19 and breastfeeding because there's not only bacteria in milk, but there are viruses and other pathogens and non-pathogens as well. And if I talk too slow, I'm going to skip through some stuff in the middle because I never know how fast I'm going to talk. But I'm going to start by talking a little bit about Idaho and where we are. We're not Ohio and we're not Illinois and we're not Iowa. People get us all confused. We're actually Idaho up in the Pacific Northwest. There's Moscow in the very top of Idaho. Idaho actually has two time zones, but we only have one area code. It's a small state. There's only 1.4 million people in Idaho. And you can see Boise, the population center is in the bottom. Idaho has loads of wilderness area next to Alaska. We're second in the nation. It's a beautiful place. Up in northern Idaho where we live on the Palouse. It's an agricultural region. And if you've heard the term Appaloosa horse, it came from the Palouse. So we're from that part of the country. It's beautiful. Looks a lot like Tuscany, actually. It's been called the US's Tuscany. It's beautiful. University of Idaho is an old university. It was actually established, this is our administration building, before Idaho was a state. So we are actually in the state's constitution because we're older than the state. I am the director of the Margaret Ritchie School of Family and Consumer Sciences, and I want to spend just 1 minute talking about that is because does anybody in here know what family and consumer sciences is? What did it used to be called? Home economics. We are a home economics school. Very good. Extra points. So we started off as the school or department of domestic sciences. This is how women across the country started going to university back in the day, back at the turn of the century. Same thing in Idaho. We were one of the first home economics departments. It was domestic sciences turned into home economics and now family and consumer sciences. There's never been an identity crisis. There's just been a crisis as to what to call it. These are just some beautiful images of home economics over the years at the University of Idaho. And it really was the original women's studies department. Honestly, this is how women went to college. We are now a complex discipline, everything from nutrition to apparel, textile, and design, early childhood. We teach people how to be K-3 teachers and FCS teachers, human development, family studies. So we're quite a diverse school. And we have a few graduate programs as well. So that's your little introduction to the University of Idaho. It's a beautiful place. If you ever get a chance to visit us, please do. Okay, so let's talk about milk microbiome. And again, I'm going to bring up this concept of serendipity because That's how we got into this. When we got into this whole project, we thought milk was sterile. Everybody thought milk was sterile, unless it was contaminated somehow or produced by an infected breast or udder, right? So we assumed milk was sterile unless it was contaminated. So I'm going to give you a little story about my research team and how we got into this. This is a picture of the McGuire-Williams research team from a few years ago. This is me. This is my better half, Mark McGuire, who's also a lactation physiologist, nutritionist, but he studies dairy cows. So we're a team, we're a research team. This is Janet Williams. She's been with us for almost 25 years. So it's the three of us. We've been doing research in this area for quite a while. We started off looking at mostly lipids. We were interested in regulation of fats and different lipids in the mammary gland and in milk. And in particular, we were interested in this one with a long name called conjugated linoleic acid. This is a fatty acid that's made in the mammary gland. It's very high in milk, surprisingly high. And so when you consume dairy products, you get quite a bit of conjugated linoleic acid. Really interesting fatty acid. The most potent anti-carcinogen naturally occurring that we know of, believe it or not. Yep. And it's in ice cream. That makes me very happy. And it's also anti-inflammatory. So we had done some studies on this fatty acid. We were interested in its anti-inflammatory effects. And at the time, we basically focused on very small scale, very, very controlled dietary intervention studies. And so in this particular study, I'll talk about it in a second. This is actually what made us realize that there was a milk microbiome. But I will tell you also, we can get interested in anything in milk. So if you have interest in any of these things, let me know. I'd love to talk about them. We've published on hormones in milk. I did a very controversial study a few years ago with Monsanto, looking at Roundup and whether its active ingredient got into milk. That was ridiculous. I mean, that got me FOIA'd and everything else. That was a mess, but it turns out it doesn't show up in milk. We've looked at macronutrients, micronutrients, oligosaccharides. This is our new one. We're looking at cannabis use in breastfeeding women and whether the cannabinoids get into milk. Very interesting. And up until about 2006, we did all these studies in Moscow, Pullman, very, very nice, easy studies to do. 2006, everything changed. And in 2006, I was at Washington State University, and I was invited to give a talk just like this at the University of Idaho, 7 miles away, in a biology seminar. And I was just giving a general overview of the studies that we had done on the Palouse looking at milk. No big deal, but it was just like this, but it changed my career. So those of you who are young scientists in here, you never know what you're going to do that's going to change your career. You just never know. So anyway, I was at this seminar and I was talking about this study that we had just done on CLA, that fatty acid, because we were interested in whether if you give a woman CLA, could she decrease her risk for mastitis? So we were interested in inflammation in the mammary gland. And so we did this study, randomized control study, 16 women, so small scale study, but very, very controlled. And so we were giving people CLA or not CLA, and we took 3 milk samples during this study. And we did a couple of things with those milk samples. We sent them to the mastitis lab at Washington State University. So they take cow's milk all the time and they culture it for staphylococcus and streptococcus and all the bad things to tell farmers whether they had mastitis in this cow. So we did that. And they got back to us and they said, all your milk samples are sterile. Nothing grew. However, we also we did smears of those milk samples. We stained them, and then we looked at those smears under a microscope, and we could see bacteria. So on the one hand, we're like, your milk is sterile, and we're like, no, it's not. We can see these bacteria. Well, luckily, in the audience was a faculty member at University of Idaho. His name was Larry Forney. He just retired. He's a great guy. He was part of the Human Microbiome Project. He did the vaginal microbiome with some other folks across the country. And so he was very aware of these new techniques at the time where you didn't have to culture your sample to see if something grew. You could look at the DNA in it. You could sequence the DNA, and then you could tell what you had in it. So it didn't rely on culturing. It relied on these really high-tech methods that we had at the time. And so Larry raised his hand and he said, Shelley, would you have commensal bacteria in your milk that maybe wouldn't grow on those culture plates? And I said, I don't know, because I don't know what commensal means. What do you mean? And anyway, he took me into a whole new area in my career, and I'm always grateful for that. So he reached out to some colleagues that he knew that were able to take our samples, and they were able to use this next generation sequencing technique that was pretty new at the time. And lo and behold, this is where serendipity comes in, because we did not expect to find this, right? Your most interesting findings are the things you don't expect. And this is real serendipity. So a definition of serendipity is finding something good without looking for it. Serendipity itself, though, I will say, will not get you where you want to go. You also have to be really curious about those findings. So this combination of serendipity and curiosity is key to science. And again, remember that as the young emerging scientists in the audience, you got to have them both. So we ended up publishing that paper. I'll go through this figure in a second, but note the date on this. This is 2011. It took us a long time to publish this paper. Anybody want to guess why? No one believed us. They said, you guys contaminated your milk. And we're like, well, we could have, but we don't think so. Anyway, now there's hundreds of papers on this topic, and so we did not contaminate our milk. So let me talk you through the results. This is the first paper that was published looking at the milk microbiome using these high-tech methods. And so each one of these, you can see S1, S2, S3, these are the subjects. So we had 16 subjects. Each woman gave us 3 milk samples. So each of these bars is a single milk sample. And then the way that you look at these is you just basically look at the color distributions. You don't have to know the details here. You just look at the color patterns. These are called stacked bar charts, and I'll show a few of them today. So for example, this particular woman had 3 milk samples that had, I don't know, 10 to 20% of this dark green. And you look down here and you're like, oh, dark green is Ralstonia. Okay, so you just look at the color pattern. So there's 2 takeaway messages from this stack bar chart slide. One is that each woman's milk samples had a microbial fingerprint. That means that each milk sample, and these were taken a couple weeks apart, had pretty much the same bacteria in it. So there was a fingerprint within each woman. However, there was a lot of variability amongst the women. And this is a very common microbiome finding, right? We're different. We all have our own microbiomes, and it's very consistent over time, okay? So this was the first study that was published using these methods. The top bacteria, I'll just tell you, are Streptococcus and Staphylococcus, which is why they thought we contaminated it. But now most studies show that these are the top bacteria in human milk and cow's milk for that matter. So this is a real paradigm shift in nutrition, right? So we thought we should be feeding babies sterile things. Turns out human milk is not sterile. We now know that these bacteria are also living and doing things, so they're not just dead. So real paradigm shift in science in this area. And I always like to show this slide or this picture as, what is a paradigm shift? Paradigm shift is when you look at the same thing, but you look at it differently and you see different things, but it's the same picture. So if you look at this picture one way, it looks like an old lady. If you look at it another way, it looks like a young lady. Can you all see that? That's a paradigm shift. Depends on how you look at it. In our case, we were looking at milk differently. We were using a new technique. Looking at the same thing, we found different results. So, and if you did a PubMed search on the human milk microbiome, this is back a year after we published that first paper. Of course, we'd been presenting the work for 5 years at that point. And then here's 2023. There's 320 papers just in 2023. So it's really changed things. And if you look back over time, we probably should have known that there was a milk microbiome, because people had been studying bacteria in milk for a very long time. And I just want to point out a couple things. In the 1950s, we knew that there was Staphylococcus and Streptococcus in milk, but we called it a contaminant. But there was a lot of evidence that it was there. We just thought it was a contamination. In 1985, we found out that HIV could be transmitted from mom to baby via breastfeeding, via the milk. So that was the first time we realized a pathogen could be transmitted. In that case, it's a virus, but we knew that there were microbes in milk. In 2004, a group in Spain, the Martinez group, really proposed that there was a milk microbiome. They were using culture-dependent techniques, and people didn't believe them, even though they were doing great work. Here's our paper in 2011 using this next-gen sequencing. That really changed everything when that paper came out. And then I put a star next to the COVID question mark, because I'll tell you that was a huge deal to try to figure out whether the SARS-CoV-2 virus could be transmitted. And I'll talk about that at the end of the talk today. So since we published that first paper, there have been a lot of studies done using these newer techniques to look at the milk microbiome. And this is just a figure out of a 2018 paper where they were comparing different findings around the world. And they concluded that, in fact, Staphylococcus and Streptococcus were core bacteria. I don't know why it's doing that. I'm going to click it over. Yeah. Yep. So that was not a huge surprise. And I will say that we find the same thing in cow's milk. So staph and strep appear, and we found it. We actually milked grizzly bears one time. They also have staph and strep. So it does seem to be a core bacterium, two bacteria that babies, infants are, thank you, are used to getting as neonates. You mean to take it down to the speciation level? They are skin bacteria. Yeah. Yep. Very much so. So some of the origin for those bacteria in milk are definitely coming from the skin. No question about it. Yep. Okay, so the next question we had to begin with is there were a few groups around the world looking at this, and we were finding different results in different countries. And in human milk research, we're always interested in what's causing variability. So that is still a big question in the field. There's a lot of variability. I showed you that in that one slide, but in fact, there's just loads of variability. So people look at the standard things that we look at in human milk, things like gestational age, delivery mode, stuff like that. This is just a nice review from 2018 kind of talking about this. But to be honest, we don't know. We don't know what's driving all that variability. I'm going to talk about a few factors today, but I'll just tell you from the start, begin with the end in mind, we don't know. We don't know. But I'll talk a little bit about time postpartum. And then, of course, the most important factor that everybody should be concerned about is maternal diet, says the nutritionist up front. I'm real interested in that. This is an interesting one. Do childcare practices and social environment impact milk microbiome? We have some data on that. Geographic location, and then other milk components, because this is a complex food. There could be interactions going on in the mammary gland. So we'll start talking about those first few things, time postpartum and diet. This is the first study that we had funded for the milk microbiome. Thank goodness for the Gates Foundation. They always seem to be at the forefront of everything. And this was a study that we did in the Moscow-Pullman area. It was a longitudinal, repeated measures human study. So we followed women and their babies over time. We studied 21 healthy moms and babies. We're always interested in healthy first before we can understand disease. So we started with 21 women. We collected multiple samples extremely carefully so that we knew that they were not contaminated at 2, 5, and 10 days postpartum, and then monthly till 6 months. We also collected other samples, infant saliva, feces, maternal saliva, and maternal feces, and I'll show you those data because we were interested in, are these microbiomes the same or are they different? We had 3 basic objectives. The first was just to characterize that milk microbiome over the first 6 months and to see if it changed. Our data show that it's pretty consistent over 6 months. So whatever the mom is producing in those first few days, she's producing the same sort of milk microbiome 6 months later. I won't show you those data, but they're published. And then the second objective was to look at diet and see if there are relationships with milk microbiome. And then third, to compare and contrast those different kinds of microbiome. We thought that would give us good information. So we'll start out with the diet. Ignore this for a second, because that's complicated if you haven't looked at heat maps. What we did is we collected really detailed dietary 3-day records at each one of those time periods. And then what we did in this data set is we averaged dietary intakes over time. So we were able to, go all the way down to how much vitamin A, how much B12, a woman was consuming. And we looked at the data a variety of ways. So we looked at these data are actually the means over time. So we would consider these chronic intake levels of different nutrients for these women. And that what we wanted to do is to correlate those types of intakes, let's say carbohydrate intake, with, let's say, how much streptococcus was in the milk. Okay, so we did a whole bunch of correlations. So these images, these are called heat maps. This is a way of visualizing a whole bunch of correlations at the same time. So here we have the different bacterial taxa. These are actually the phyla of the taxa on the x-axis. And on this one, we have energy yielding nutrient and nutrient classes. So these are things that give us calories, okay, on the y-axis. And then the way you interpret this, let's take energy, for example. We were able to estimate kcals per day per woman. And then if you come all the way over here, I want to know, how does that correlate with percent firmicutes in the milk? So if there's, in this case, there's a red rectangle, so that means that they were positively correlated. If it's a blue rectangle, it means they were negatively correlated. Okay, so there's a take home here. If you look at this column and this column, you can see a pretty strong pattern in that, in general, the more calories a woman consumed, regardless of where it was coming from, carbohydrates, protein, fat, she produced milk with higher percentage firmicutes. And she produced milk with lower percent Bacteroidetes. Now, these are necessarily inversely correlated because the way that we look at microbiome is all on a percentage. So if one goes up, something else goes down. Yes?
Have you ever looked at the actual metabolites or nutrients in the milk itself?
Yes, I'm going to get to that. Yeah, that's the right question, actually, because it's probably what's going on in the mammary gland. Yeah, could be other things too. So this is the first indication we had that at least there's correlations. Correlation doesn't mean causality, so you don't know if this is causal, but this is pretty strong to find patterns like that. We also looked at, for example, individual amino acid intake. So these are the building blocks of protein, so alanine down to valine. And again, we have the different taxa on the x-axis. And without going into detail, you see beautiful patterns here. We don't really know the reason for this, but this is pretty striking. You rarely see heat maps that have this strong of patterns. And we also looked at the micronutrients, and we found some interesting relationships. So for example, vitamin E intake was positively correlated with firmicutes, negatively correlated with a couple others. And these are all adjusted for multiple comparisons. So it was an interesting first look at relationships between maternal diet and milk microbiome. Are they causal? I don't know. Are they the same in other populations? We don't know, and nobody else has done this sort of analysis since we've done it. It's really a need that we have. Can we shift milk microbiomes with diet? We don't know. We definitely need to do some controlled dietary intervention studies to look at this. Okay, so you might be wondering, how would maternal diet impact milk microbiome? I have 3 proposed ways that could happen. I'm just going to go through these. So let me just, so here, these are the mammary glands, right? This is the GI tract, the gut, and then that could be a breast, but I put a carton of milk there just because, whatever. And these are bacteria. So I'm just going to kind of illustrate how this might work, these 3 different ways. So the first one has to do with consumption of probiotics, so living bacteria. We consume these when we consume things like yogurt. I think it's quite likely that, if you consume a living bacteria, that it could go to your GI tract, and there's evidence. We're going to talk about this in a couple slides, and it could get trafficked to the mammary gland and put into milk. And like I said, there is evidence that this happens. Secondly, let's say you consume a diet that's high in vitamins, okay, different vitamins. That goes into your GI tract. That feeds the bacteria in your GI tract, okay? Then a differential set of bacteria would get trafficked to the mammary gland and get put into milk. Or thirdly, and this is what you were talking about here, if you consume, let's say, a high nutrient diet, those nutrients are going to get in the mammary gland, and they get into the milk, and that could, in the mammary gland, change the mammary microbiota, and then that would change the milk. Yes.
I wonder maybe this bacteria must go to mammary glands through blood. So why these people not getting infected in the bloodstream?
Because we think that they're protected in dendrocytes and other immune cells that are actually sampling the GI tract and trafficking safely to the mammary gland. That's what we think. It's a good question. So again, dietary intervention studies are needed. That's kind of my bottom line for everything. So the question is, how are they getting to the mammary gland? And we don't know for sure. There's growing research on this area, but there's two general ways that we think probably the majority of them are getting there. And these are just some images from a couple of reviews. So here you have a mammary gland, and we call it baby backwash. There's some beautiful studies. You've probably never thought of this, but when a baby nurses, the baby consumes the milk, and then when they let go and that back suction happens, whatever's in the baby's mouth goes back into the mammary gland. And there's beautiful images of this using, I'm drawing a blank because I've been awake for way too long, ultrasound to look at this. So we call this baby backwash. Think about this beautiful idea. It's a fairy tale at this point, but I think it's probably true. That baby is coming into contact with all of these environmental microbes. Everything that the baby comes into contact goes into the baby's mouth, then goes into the mammary gland. The maternal immune system can then respond to that in an appropriate way and help the baby learn how to respond. Baby backwash. The other mechanism that I just talked about is this enteromammary pathway, where we think that bacteria in the GI tract are being sampled and then trafficked to the mammary gland. And animal data show very clearly that this is happening, especially in late pregnancy and during lactation. And I just want to point out, this is a beautiful review that goes into a lot more detail about how this can happen and other environmental factors that might impact this, published in 2020. So please look up this nice review if you have more interest in this. Okay, so I'm going to come back to that Gates study that we did. So we've talked through objective 1 and objective 2. Objective 3 was to compare and contrast those different microbiomes. So the question was, is the infant's fecal microbiome the same as the milk microbiome, et cetera? So this is called an NMDS plot, and I'll talk through what this means. So if you think about it, we analyzed all these different sample types. Each one gave us, 50 different bacteria, percent this, percent that, percent, and we didn't want to look at the individual bacterial taxa. We wanted to look at the whole picture. And the way that you do that is you do these plots. They're called ordination plots, where you take, let's say, a fecal sample that has 50 different kinds of bacteria, and you summarize that data into one dot. And that's how you do an ordination plot. So each one of these dots on here, let's say I'm up here in maternal saliva, which is this pink one, each dot represents the bacterial community of a woman's saliva in one dot. Okay, so if the dots are close together, it means that the bacterial communities are similar. If the dots are far apart, it means that they have different bacteria in them. Okay, so it's a way of looking at this in a picture, which I like. I like pictures. So here's the bacterial community structure of maternal saliva. Here's infant saliva. So very similar. Maternal and infant saliva have similar microbiomes. Here's milk right here. Here's infant feces, and here's maternal feces. So, you can see here that these are quite different. The milk microbiome it's a different community structure as the maternal feces. Now, you might look at this and say, well, oh, I'll get to that. So we did this across all of those days. So here's day 2 postpartum, here's 6 months postpartum, and you can see that these differences are already there at day 2. So this is very robust over time. So you might look at this and say, the milk is completely different than maternal feces. That enteromammary pathway must not be true. But we don't look at that at all that way, because if you think about it, you take a fecal sample, does that represent what's going on in the upper GI tract? Not at all. We know that microbiome changes throughout the GI tract. So just because the feces are different from the milk doesn't mean that, let's say, the GI tract wasn't sampled, let's say, in the upper part, in the duodenum. Okay, so this doesn't negate the enteromammary pathway that way, but we used a statistical technique called canonical correlations, where you can actually, again, take that really complicated data, turn it into one dot, and then just compare these dots like you would do in a normal correlation plot. Okay, and look at correlation. So what we're looking at here are correlations between variability, not actually correlations between community membership. So what does that mean? It means that if something changes in the milk microbiome, let's say Staphylococcus, then something consistently changes in the milk, but it might be a different bacterial type. So that's what these canonical correlations are. And we get these beautiful correlations that you rarely see in biological samples. So if the milk microbiome changes in some way, it is correlated to what's going on in the maternal fecal samples. And that's a really high correlation, but look at these. Here's a correlation between infant oral microbiome and milk microbiome. This supports the idea of baby backwash. Of course, it could go either way, right? Because the milk microbiome could be seeding the oral microbiome. But I will tell you that there's been a beautiful study done where they actually took mammary secretions before birth. And even in primiparous women had never had a baby, never breastfed, those mammary secretions already had the microbiome that we would expect and seemed to be seeding the infant mouth. So which is coming first, the milk or the baby's mouth? Probably the milk. And then here we have relationships between milk microbiome and infant fecal microbiome. So definitely correlations going on here. Okay, so we've talked a little bit about time postpartum, maternal diet. I want to talk a little bit about this social interactions. I think this is fascinating. So this is aside from my husband and Janet, my favorite colleague, Courtney Meehan. She's an anthropologist, a biological anthropologist at Washington State University. And she had been studying family structures and childhood in the Central African Republic. And she was interested in breastfeeding. And we got together and I said, hey, can you bring some milk back from the Central African Republic? And she's like, well, okay. But I study hunter-gatherers, so I have to go find them first. And there's no refrigeration, so I'm going to have to like bring in a generator. And she did all this. And she went and she studied women and babies in the Central African Republic. And she collected a whole bunch of data, and I'm not going to go through all this, but what we looked at is the relationship between how these babies are cared for, because in these hunter-gatherer populations, they have what's called allo mothers, right? So the moms are busy. Everybody takes care of everybody's babies, and family structures are much tighter that way. And what she was able to show, without going through the detail here, is that the more people that took care of a particular baby, the more diverse that baby's that milk microbiome would be. Okay, so diversity of care, that baby's coming into contact, let's say with 20 people in a day, that mom is going to have a greater milk microbiome diversity than some baby who was only handled by 2 people. And we found that very interesting. Very, very interesting. And we have, right before COVID, we launched a project like this in the United States. this was not fun to do during COVID, but we're actually just starting to publish some of the data. I'm not going to go through the details, but we studied 51 healthy moms living in the Moscow-Pullman area. We took all these intensive observations. So we were there for 3 days from 7 a.m. to 7 p.m. and actually did observations every 30 seconds. The data here are so rich. And then we took milk and we looked at the milk microbiome and did regular microbiome analyses. And this paper is just coming out. And I'm not gonna go through all the details, but I want to point out some interesting relationships we found. We found that moms that worked outside the home had greater diversity in their milk microbiome. Kind of interesting. What we didn't expect is that frequency of non-mother physical contact was actually inversely correlated with milk diversity. That's opposite of what we saw in the Central African Republic, but we have very different things going on in these two populations. We also found that the more frequently a mom breastfed, the lower her milk diversity was. We don't understand that. Doesn't make sense, but we'll figure it out. And then we found also the greater the caregiving network was. So this is a little bit different. This is a different metric than this one. Here we did find the same relationship that we saw in the Central African Republic. So the bigger the network of people taking care of the baby, the greater the milk microbiome. Okay, what about population differences? Well, we launched a study. I was told I'd never get NSF funding. And here's an example of don't believe what people tell you. Because the NSF actually funded this project. We were interested in what is normal milk microbiome. And our hypothesis was that there is no such thing as normal, depends on where you are. So normal shifts depending on where you are. This was a cross-sectional, cross-cultural epidemiologic study. We had 11 cohorts in 8 countries around the world, resulting in around 400 mom-baby dyads. We studied them between 1 and 3 months postpartum. And the beauty of this study is that we standardized everything. So we even sent collection kits that contained diapers, contained gloves, contained the same washing supplies, everything. So we standardized everything, and then we analyzed everything so that we could be really confident that if we found differences, they were real and not just due to methods. We also collected infant feces and maternal saliva in this study. This is the Inspire team. Obviously, this is a lot of fun. This was before Zoom. We did this before Zoom. We never had a single meeting. It was awesome. Wow, we did that. Anyway, we've published quite a few papers on this study, and this is kind of the results of the main paper looking at the milk microbiome. So these are these stacked bar charts again, so you just look at color patterns. Here we have rural Ethiopia, urban Ethiopia, rural and urban Gambia, Ghana, Kenya, Peru, Spain, Sweden, US California, and then US Washington. So these are the milk microbiomes of all these regions. To begin with, you can see that there's a lot of variability, right? Sometimes they're the same, sometimes they're different. But a couple things I want to point out. In rural Ethiopia, the top bacterium was Rhizobium. Anybody, does that ring a bell? Rhizobium? Nodules, lentils, legumes? No, it's a soil bacterium. The staple food here is a fermented root. So they're consuming a lot of rhizobium because they're fermented in the ground, actually. So we think that's where that's coming from. And then interestingly, in Ghana, some of the top ones included Klebsiella. These are healthy women. There's no problem there at all. So again, another example of what you think is, you know, don't fool yourself that you know what healthy is, because this is not a problem there. They also had a lot of lactobacillus. They eat a lot of fermented dairy products. And in US California, one of the top ones was Escherichia shigella. We can't tell the difference in our analysis, but those are usually not considered to be good bacteria. But again, healthy moms, healthy babies. When we did canonical correlations here, again, with all these countries, and this is between infant fecal microbiome and milk microbiome, again, we find a really nice correlation. So even though the bacterial communities are different, their variation is correlated. So we really think, and again, this is fantasy at this point, but we need to do some more studies. We really think that this milk microbiome is optimized for a location, and that it helps optimize the infant's GI microbiome for that location, and that this variation probably is important, would be our guess. We wonder if we can optimize an infant's GI microbiome by affecting the maternal microbiome via diet, via probiotic drops. We don't know, but it's an area of real interest to us. I'm going to skip the next two. This actually gets back to milk, other milk components. Who brought that up? You did. So I just point you to this paper. We actually did look at that. These are some other milk components. What's highly correlated are HMOs. So these are complex carbohydrates. And so these are just correlations within milk. And we also found similar correlations with the baby's fecal samples. Again, I'm not going to talk too much about that. And actually, I think I'll skip this too. Okay, so the question is, so what? I don't have much on this. We don't really know impacts on infant health or maternal health at this point, but we are doing a study with a colleague of mine at Johns Hopkins. You might be familiar with this study. It's the Jivita trial in Bangladesh, where they've looked at a lot of infant outcomes, infant growth. In this particular study, we have samples from 119 exclusively breastfeeding moms and babies. We have milk and infant anthropometrics at 3 months of life. And our hypothesis was that variation in the milk microbiome would be very related to variation in infant anthropometrics. And interestingly, so here's Shannon diversity. So this isn't just a measure of how diverse the milk microbiome is. And at 3 months, we're surprised to find that increasing diversity is related very strongly to increased length and to increased head circumference. I mean, we were hoping to find some relationship, but these are pretty strong, interestingly. And we also find the same thing with richness in the milk. So that's how many different types of bacteria are there. We haven't published this yet. We're still working on the values. If you look at the individual bacteria, what we find is that as streptococcus goes up in the milk, weight goes down, length goes down, and head circumference goes down. And you find the opposite with staph and Corynebacterium, depending on the variable. So we're still trying to understand these data, but these are the first that actually relate variation in milk microbiome to infant health in this way. Okay, and lastly, in my last few minutes, I want to talk a little bit about COVID. I think this is probably the most important work we'll ever do. Yes.
Yeah, there's been a lot of work with the microbiome to behavioral.
Yep.
Much later. Do you have any intention of doing more?
Yes, absolutely. That's our next NIH grant if they would just fund it. Yep. That's the obvious thing to do. Yeah. Yes.
What about comparison between formula fed versus breastfed?
Well, there's a lot of those papers out there, the comparisons. So we know that breastfed kids get sick less and grow differently. But to be able to say it's oh yeah, there's big differences between breastfed and formula fed babies. We always assumed it was other things in milk, but I'm 100% sure part of it is the milk microbiome. Yeah. We assumed it was the HMOs and the oligosaccharides and lactoferrin and iron and things like that. But okay, so I'm going to talk a little bit about this. This is a fun story. So the question is, can SARS-CoV-2 be transmitted from moms to babies during breastfeeding? As I mentioned before, we knew that HIV could. And so at the beginning of the pandemic, and I'm just going to set the stage here because this went really, really fast. Remember, end of February, first deaths due to COVID were announced. They were in Washington State, so right next door. You know, a couple days later, we reached out to WHO because I know the guy who's in charge of all the breastfeeding stuff, Larry Grummer-Strawn, and I said, this is going to be an issue, Larry. What's been done to see if this virus can be transmitted? And he said, don't worry about it. It'll be fine. Nobody's going to worry about it. And I'm like, yeah, you're not right. So a couple of weeks later, WHO characterized this as a pandemic. Right about then, we started coalescing a group of human milk researchers from around the world Thank God for Zoom. This is the first time we really used it. This is a beautiful outcome of a society, the International Society for Research in Human Milk and Lactation. We're a tight group, and we all said we need to hit this head on and figure this out because this is going to be an issue. So March 30th, the WHO came out, they're like, don't worry about it. Here's their thing then, like, don't worry about it. Just keep breastfeeding, it's going to be fine. But so then we're like, this is not going to be fine. We started writing a formal lit review to see what we could find on it. April 2nd, American Academy of Pediatrics released their initial guidance, which basically said, yeah, we don't know. Yeah, there's no evidence that it can be, but we're just going to suggest that we separate moms and babies if the mom is infected. If the mom wants to breastfeed, she should just pump somewhere else and give the milk to a healthy person to feed the baby. That meant that when a woman went into the hospital in labor, and even if she was feeling fine, they tested them all. Remember how horrible this was? And they found out she had COVID. That baby was delivered and separated from the mom for 2 weeks. So this was a nightmare. Anybody here have a baby during that time? Because this was a nightmare. Yeah. I'm not going to read all this stuff, but AAP advice was separate. CDC came out, basically said the same, like, yeah, you should just separate from your baby. This was awful. Remember, nobody was getting tested. You didn't even know if people were sick. So who was supposed to feed your baby? Yeah, express breast milk should be fed to the newborn by a healthy caregiver. We couldn't even test people. Yeah. And then April 6th, the New York Times puts this thing out, and they basically say, yeah, temporarily advise separating moms and babies. So that initial response from WHO saying, don't worry about it, was not true. This basically threw the world into a tizzy. So we wrote this first review on SARS-CoV-2 in human milk. What is the evidence? Again, this is kind of an international group that put this together, and just kind of put this in perspective. We started writing it April 1st. Okay, we started writing it April 1st. We sent it in April 7th. Remember, we didn't have anything else to do. At this point, there were 9 publications on the topic looking at 16 women, 20 milk samples. They were all negative for SARS-CoV-2, but they didn't give us any methods. We had no idea how the samples were collected, how they were analyzed, whether they validated the assays for human milk. We're like, yeah, we don't believe any of that. The paper was accepted May 5th, but by May 11th, there was like a paper a day coming out on this. At that point, people had analyzed 80 milk samples. 3 of them were positive. One of those papers was published in the Lancet, right? So now we're game on. We have to figure out if this is true or not. But we didn't trust any of these papers because they didn't tell us how they collected their samples or analyzed them. So we were lucky. We got funding from NIH NIH, that would have been 12 months later. But we got immediate funding from Gates Foundation and from the NSF. We ran a prospective longitudinal repeated measure study, 64 moms and babies. The moms all had COVID. We lived in Moscow, Idaho. We didn't even have any cases for like 5 months. So we had to do this around the country. So they were recently diagnosed with COVID-19. To get these samples to know that they weren't contaminated took a lot of work, right? So we put together all these kits in Moscow, Idaho, under aseptic conditions, wearing PPE, so we knew we weren't contaminating them. And we also collected breast swab samples because we wanted to know if there was virus on the breast. So anyway, very standardized. And then we validated the assay, the CDC assay for human milk, so that we knew we trusted it. So we were in weekly contact with the CDC, the WHO, and the Bill and Melinda Gates Foundation as we were doing this, so they knew the data as it came off. We were confident after the first 18 women that we collected 37 samples from. There was no SARS-CoV-2 RNA in any of those samples using our methods, which we trusted. So we published this data. Interestingly, 35 of the breast swabs that we collected from the breast prior to cleaning the breast actually had evidence of the virus on it. So there was virus on the breast, but it wasn't in the milk. So that's actually an important distinction. However, none of these babies were sick. We had no evidence that this was a problem to the babies. And after she cleaned the breast, then there was no evidence of SARS-CoV-2. So we continued this study. We recruited another 47 participants from 24 states. This was actually a multi-site study. Thank goodness, because as I said, we didn't have any subjects in Moscow. So we worked with people all over the country. In the end, we had 316 milk samples collected from 64 women, absolutely no evidence of SARS-CoV-2 in any milk sample. Again, presence on the breast, but it was washed off, and there was no evidence that these babies were sick, which was really important. And on top of that, the milk contained neutralizing antibodies to the virus. So not only was the virus not there, but the specific antibodies were. So in summary, cute baby, right? Human milk has an inherent microbiome that varies among women, and it varies globally. It's relatively stable within a woman. We think it's probably customized to a culture and an environment. May educate the baby about what microbes to tolerate, may also educate the baby how to fight off the bad ones. We think it may optimize, there's a lot of mays here, GI microbiome for long-term environment. Variation in the milk microbiome is related to, and again, I'm being very careful here, not to say it's causal, maternal diet, childcare practices, social environment. I didn't show you, but also the study we did in the United States, the biggest determinant of diversity is pets in the home. So the more pets a woman has in the home, the more different kinds of bacteria she has in her milk. Related to maternal and infant fecal microbiomes, salivary microbiomes, and I didn't show you this data, but it's also related to breast inflammation. Sometimes there's transmission of pathogens, but we were lucky in this case with SARS-CoV-2 that is not transmitted via milk. And there's a lot to learn. And then my final slide is don't forget serendipity, because this was all an accident, and it was a good accident. And we were smart enough and curious enough to move forward with it. And then I have a plug. So we just got a big grant to establish an NIH Center of Biomedical Research Excellence in Nutrition and Women's Health in Idaho. And women's health is not so good in Idaho, so this is a big deal for us. But we have our first hire advertised. So if you know of anybody at the associate professor or professor level who wants to do nutrition and women's health, we're looking for somebody to fill that role. So let me know. And thank you. And I think I have 1 minute.
That's okay. Thanks so much. Some questions online. I'll just share one.
Yes.
One question was, is it possible to produce infant formula enriched with microbiota?
Yeah, that's a good question. So what does this mean for infant formula? What it doesn't mean is let's add streptococcus and staphylococcus. That's what it does not mean. Now, people, another way to look at that is people are actually now getting microbial communities from women's milk and then inoculating, let's say, donor milk, so that that milk has what the baby would have gotten had the baby nursed from the mom. That, I think, is a more viable and interesting solution. But what I do worry about with formula is they're just going to start adding bacteria. I don't think that's a good idea.
And actually, I'm going to take a question before I call on you. I can't resist. When you showed us the graphs of the comparing the women in, I think, Washington and Idaho or California and Ghana and various places, I guess I would have expected in a non-agrarian area, I don't know if people were in cities or what, but you'd have a lot of variability. I mean, even though you may live in a city like Seattle or Moscow for that matter, the diets that people have can be extremely varied. You know, people are vegetarians, et cetera. So what sort of variance was there within those areas?
There's variance. Yeah, there's variance, but on average, they're different, but there's a lot of variance. One thing I will point out from that slide, which I find really interesting. So if you look at the two Ethiopian sites, very different microbiomes. One is rural, one is urban, very different. If you look at the two in the Gambia, also one very rural, one very urban, exactly the same microbiomes. Differences in, does anybody know Andrew Prentice in that group in the Gambia? That's who we were working with. So they recruited women in Kenaba, which is a rural site first, and got the milk there. Then the way that it recruited women from the urban site is they asked the Kennebah women, hey, do you know anybody who lives in this site? And they all suggested their sisters and cousins who had moved there. So those women had grown up in the same rural site and moved. And it makes me really wonder if it's really early establishment. Yeah.
Okay, we got a row of student hands up there.
Hey.
Go ahead, question.
My question was about antibiotic use. I know that if you overuse them, you can deplete your gut microbiome, and I was curious how that can impact the mother.
So we've had a few cases like in that longitudinal study where the woman did use antibiotics, and in our studies, they use antibiotics, we kick them out, but we analyze the milk anyway, totally got a monoculture. So when the mom used antibiotics, her milk microbiome shifted immediately.
Pass the mic. Oh, there you go. Good.
Great talk. I wanted to ask about how closely the infant fecal microbiome related to the breast milk microbiome. There's been some work on the microbiome that infants develop through their exposure to the vaginal microbiome during vaginal birth that would obviously differ from babies born by cesarean. So I was wondering if you noticed any patterns in how closely the infant fecal and milk microbiome related in vaginally born children versus cesarean.
So we and most people don't find differences in the milk microbiome, depending on how they delivered. There's a couple studies that suggest it might, but yeah, in the infant fecal, also that's controversial. Some studies show a difference and a lot of studies don't. So I think the word is still out on that. The milk microbiome and the infant fecal microbiome are quite different. They're different community structures, but their variation is highly correlated. But we don't really see much difference in cesarean versus vaginal, which kind of surprises me, but everybody gets antibiotics these days. So that's what we need to find. We need to find really different groups. We need to find vaginal deliveries that had almost no interventions and then compare that.
Okay, we're on a roll with student questions. There you go. One more question. Go ahead.
Yeah, I don't know if you are willing to kind of extrapolate a little bit with
Never.
Right, milk being like the only food for humans that's been like tailored for millions of years to support health.
Yep.
And you're finding that a rich microbiome is important later in life. Now we're eating foods with increasing degrees of ultra processing, and shelf life is really important, and sterility for storage. Is this kind of like I tell that, it's development, you're seeing a lot changing. Like, is this also potentially affecting adults? We just don't realize it yet.
Probably. Probably. I mean, I think that these probably have long-term effects. We know that the GI microbiome is set up in the first couple of years and it's hard to change it after that. I find it almost impossible to believe that the milk microbiome isn't influencing that, right? So I think you're on the right track. Do we have evidence for it? No. Job security. Do milk microbiome. There's so much to learn.
Okay, time for one last question. Well, I see two hands. Go ahead. You grab a microphone.
I was really interested in the correlations you're drawing between the diversity of the microbiome and then some of those biometric measurements of the newborn. And I wondered, kind of beyond just a simple correlation, if you have enough data to be able to control for any kind of confounding variables that could impact that development, for example, whether or not a baby was born prematurely or.
So these were all full-term, relatively healthy Bangladeshi kids. I mean, they're born into difficult situations, but they're relatively healthy. So, and they're all vaginally delivered. So it's a pretty tightly controlled study. And I'm trying to think what else we could control for. We did control for estimated, they were full term, but we did have some estimates for, gestation length, nothing, but the data, it's Bangladesh. I mean, nobody knows when they got pregnant. So that's kind of hard. So we don't have a lot.
Okay, now one last question. There you go.
Not sure if I understood correctly. Did you say, this is the first part, did you say that, yes, the leading theory is on the backwash?
The backwash and the intramammary pathway.
Okay, so both.
Both. Yeah.
Okay, because what didn't make sense to me was if it was consistent, then on day 2, an infant's backwash is going to be hardly they've hardly been exposed to
Mhm.
anything else. And then at 6 months, they've been exposed to a ton of factors, right?
Yeah.
But
that's a
it,
good-
but it
that's a good
stayed
point.
consistent from,
Yeah.
that day 2 to 6 months.
So they're not that ambulatory yet. Some of them are and some of them aren't. It's a good question.
Right.
It's a good question. I'd like to actually look at it further out, which nobody's looked at yet. It's a good question.
I was
Yeah.
thinking like daycare and that type of thing, or sibling, I mean, so many,
Nobody's
right?
looked,
Like,
I know, you're right. They would come into, so the other way you can look at it is the mom's already been exposed to all that stuff for years and years. And so baby's just getting exposed to what the mom got exposed to. That study that we did in the US, we meant for that to be a daycare study. So we had daycare kids and home kids, but then COVID happened and all the daycares closed. So we had to punt. But I agree with you. I agree with you.
Okay, please join me in thanking Dr. McGuire.